Assalamualaikum and good morning to everyone. So today we are going to present to you on a research paper about a universal test for the forensic identification of all main body fluid including urine. But before that, let's watch this short video. Of biological fluids at crime scenes is a crucial step in forensic investigations as it can significantly influence the trajectory and outcome of a case. Among the common biological fluids such as blood, saliva, semen, sweat and vaginal fluid, urine has historically been overlooked in many universal detection methods despite its relevance in cases involving assaults or sexual crime. Traditional forensic approaches involve a combination of presumptive and confirmatory tests. Presumptive tests such as those using alternate light sources, catalytic color reaction and enzymatic assays offer a rapid screening capability but are often body fluid specific, which is prone to false positive and negative and can be disruptive to the sample. Confirmatory tests including immunological assay, microscopic analysis and chromatographic technique, while more reliable, require a sophisticated instrumentation, trained personnel and are generally limited to the laboratories. This study presents a universal non-destructive method using Raman spectroscopy for identifying 6 major body fluids. The technique achieved 100% validation accuracy on 82 donor samples, highlighting its strong potential for forensic application. So this research came up with a quick and non-damaging way to identify the body fluids using a light-based type called Raman spectroscopy. It works perfectly on all 6 types of fluids, making it super useful for a real-time forensic work. The researcher developed a reliable and non-invasive technique using Raman spectroscopy to detect the 6 body fluids. With 100% accuracy from 82 samples, it shows a great promise for use in forensic investigation at the crime.
Next, I will briefly present the methodology for handling and preparing urine samples used in a forensic body fluid analysis study. A total of 27 human urine samples were acquired from BioIBT. These samples were purposefully selected to ensure diversity in sex and race with donors ranging in age from 13 to 65 years old. To preserve the integrity of the biological material, all samples were stored at temperatures below -20 degrees Celsius until they were ready for preparation. For consistency, the sample preparation protocol followed the same standardized method. First, the frozen urine samples were thawed at room temperature. Next, they were homogenized using a vortex mixer to ensure even consistency throughout the sample. Then, 10 microliter aliquots of each sample were deposited onto microscope slides that had been covered in aluminum foil. The foil served to reduce substrate interference during spectral analysis. Finally, the prepared slides were dried for at least 24 hours at room temperature before any spectral data collection took place.
Next is instrumentation. All Raman spectra were collected using an in-via-Raman spectrometer using Wire 3.2 software and equipped with a Leica research-grade microscope. The microscope was outfitted with a 50 times objective and a prior automatic mapping stage which was used to collect spectra from various points of stain. A 785 Nm wavelength laser source with 130 mW of power was used for excitation. At least 18 spectra per sample were collected by selecting point in grid pattern with 2 times accumulation at each point. The spectral range used for this study was about 400 to 1,800 cm.
Now, let's look at the spectral preprocessing steps used in the analysis of urine samples. First, the raw Raman spectra were imported into MATLAB version 2017B and processed using the PLS toolbox. To correct the baseline, we use automatic weighted least squares with a polynomial order of 5. The spectra were then normalized by total area. This step helped improve comparison of spectral shapes and relative peak intensities across different body fluid. Before moving forward with modeling, we min-center the data. We also remove any spectra that were affected by cosmic rays to ensure the accuracy of the results. Next, we split the pre-processed spectra into two main sets. The training dataset included 22 donors and 419 total spectra, while the test dataset included 5 donors and 86 spectra. These new datasets were combined with the existing calibration and validation datasets from the previous study, which already included 5 body fluids. This brought the total number of donors in the training dataset to 82. Finally, we use this expanded dataset to train the new SVMDA model to classify 6 different body fluids. Variable selection for this model was done using a genetic algorithm. The model's performance was then tested using a new validation dataset that consisting of spectra from 20 donors.
Overview of SVMDA approach. SVMDA or support vector machine discriminant analysis is a popular classification method. Some advantage over other classifier is it robust with limited sample size, it produces reproducible result with same parameter, kernel tree enables separation of nonlinear data in a higher dimensional space. It also helps reduce overlifting when using a subset of selected variables. Some update SVMDA model with genetic algorithm. GA selected 665 informative variable. It used 7 latent variable to identify all major body fluids in a single step. RBF or radial basis function kernel use. Kernel parameter optimized using grid search with cross-validation. Venetian blind cross-validation assess model performance. External validation done using 20 separate donor sample. GA identified key Raman spectral region for each body fluid. Next is model training on N multi-class classification. New training dataset about 82 donors. Goal: classify 6 different body fluid. 1 versus all approach used for multi-class SVM. Each SVM trained to distinguish one fluid versus all others. Model trained using GA selected variable from previous work. Next is cross-validation and evaluation. It used Venetian blind cross-validation. Dataset split into N subsets. Model trained on N-1 subset test on the remaining. It repeats N times so each spectrum is validated once. Confusion metric used to evaluate model accuracy. No probability threshold use classification based on highest probability. Next is PCA analysis. Non-negative PCA conduct on urine spectra to examine spectral variation. Some of the tool used is PLS toolbox.
I'm going to talk about the result analysis based on Raman spectroscopy for this research topic. Why do we need to improve our method using Raman spectroscopy is to identify body fluid for forensic cases. We have the models can already identify 5 body fluid, but which is blood, semen, saliva, sweat, and also vaginal fluid. We are adding on urine on top of that. So why does Raman spectroscopy is used? Because it's non-destructive, fast and accurate, and it is able to detect the unique chemical features in each body fluid. So as we can see in table 2 and 3, we are shown the peak and also the PCA based on what we have detected. So the most important peaks is 1013, which is the strongest signals, and it comes from urea, which is the main compound in urine. And we also have 1175, which is also come from urea with a functional group of NH2 rocking movement, and 1541 and 1649, which is linked to citric acid and uric acid. This shows the important peaks, shows that it's reliable and strong peak that can identify with confidence that it is urine. And then we have something called PCA, which is principal component analysis, which shows the other important urine compound such as hippuric acid, creatinine, phosphate, and also urea. So it confirmed that urine has a unique chemical mix that makes it easy for us to identify it. So our model could be updated very well, as we can also recognize from 5 body fluid to 6 body fluid. So by adding on urine, we also improve our working skills in real life during forensic investigation. Next, I'm going to talk about the SVMDA models, which is models that are able to classify the accuracy of the urine. So we have two types of validation: internal validation and external validation. So internal validation, we tested on 419 samples on the tested data or training data, and we are able, the model are able to identify all 419 samples, which make it 100% internal accuracy. And we added on 86 new samples for external validation, and all 86 new samples are able to identify by the models also, make it also 100% external accuracy. But the overall result were 99.4 accuracy during cross-validation when testing on different part of the training data, and 100% accuracy on the real new data. This shows that our models is very reliable, consistent, and also excellent at detecting urine. It also make it easy for us to practice it in real life, such as in lab or crime scene, make it our work faster and easier and more trustworthy.
In conclusion, this study created a universal test to identify a 6 body fluid. It shows that 99.4% accurate during testing and 100% correct in final validation. This helps solving the crime by identifying body fluids quickly and reliably.